Okay, so we're going to come back with cell structures, talking more passive versus active transport, kind of just coming off of our cell membrane talk that we had, just overall function and kind of the characteristics of the cell membrane. Remember, we go back and talk about the ability to move certain substances in and out. We'd said it's selectively permeable, amphipathic, and there were some other characteristics that the membrane also had, but I think probably one of the most important ones. Yeah, I can't even say that either because when you start looking at cell signaling and cell communication, that's pretty important, obviously. But just the one I think is probably more notable is the whole movement of substances across. So we kind of go back to it being amphipathic, and you've got the polar heads and the nonpolar tails. You've got the hydrophilic heads. You've got the hydrophobic tails, and just again how polar molecules can't easily get through, or if they're too large, they can't get through the membrane. They're going to need those transport proteins to get through. Anything that generally is, in this case, hydrophobic, if it's nonpolar, can slip through the cracks generally to get through. But either way, the two different types of transport and I would actually say this is probably another theme that we're going to talk about quite a bit throughout the course of the year, that you guys did talk about the course of the year, because this doesn't just pertain to the movement of water and molecules across the membrane as they're needed. For the most part, as we start looking at transport, a lot of it's just going to be passive. Basically, our cells need something. Molecules are going to find a way to get through. Whether diffusion is going to kind of take over, and eventually, molecules will make their way in. Whether if they're too big, obviously you've got the protein channels. If they're too small, they're going to work obviously through the cracks of the membrane of the phospholipid bilayer, and that goes both ways. And that's basically say if there's an excess amount of something, that's going to work itself back out again. And so our body has that kind of natural ability to remove and to add and to take in those certain molecules when it's needed. And that's a prime example of passive transport. And water was kind of the major example that we ended up using. We talk about these hypertonic and hypotonic and isotonic environments based on the environment that the cells are found in, and how that can change and what it can ultimately do to the cells. And so we go back to the luteal leaves that we did, and here's an example of a plasmolyzed cell to where the water has been taken out of the cell. The reason why it's been taken out of the cell is because we put the plant in a saltwater environment. So naturally, the water is going to try to make up for that concentration. All right. An example of what we call a hypertonic environment, into where basically salt water is hypertonic, large amount of solutes, water is going to leave. It's going to try to make up for that change in concentration that it was put in. We take a look at these cells over here. They're turgid, right? Turgid because we can see the chloroplasts are kind of strung throughout, pushed all the way throughout the edges. The central vacuoles are there, big pockets of water we're seeing sitting in there. And so an example of a hypotonic environment, because what's occurring is that we're putting those cells in a freshwater environment. The cells themselves have a higher amount of salinity on the inside compared to what's on the outside. So now water is rushing in, and it's going to create basically a turgor pressure on these cells, which is kind of where ultimately plants want to live and where they want to be. Now, if we talk the human body itself, talk red blood cells, a little bit different. I mean, not different in terms of the plasmolyzed, not different in terms of the turgid and so on down the line, but it's basically where we want to be. Most of our cells, when we're talking red blood cells, want to be isotonic environment. We want to basically be the equal amount of solutes on the inside and on the outside, where plants want to basically be in that hypotonic environment at all times to maintain that turgor pressure. But us, we want to maintain an equal concentration, which is why you're going to see a lot of these, again, molecules have the ability to slip through and flow through by the process of diffusion, passive transport, and work their way into the cells. Again, if we put into a hypotonic environment, water is going to go in. It's going to eventually make that red blood cell swell up, and then eventually could lead to that cell bursting or apoptosis, so we call it. Right, and obviously hypertonic leads to a plasmolyzed cell. Again, you put your cell into eat a lot of salt, eat a lot of sugar. Eventually, what's going to occur is that cell is going to end up losing some of that water, and to become plasmolyzed, it's going to shrivel up and so on. Still maintaining somewhat of a structure due to the cytoskeleton you're going to find on the inside, but it's definitely going to change shape and lose its overall shape that we see here with the isotonic environment. So again, we kind of go back to the plants. What are, if they live in a hypotonic environment, how do they maintain so much water Remember, they got contractile vacuoles. You've got single-celled organisms, protists that have contractile vacuoles that'll pump water out. Not plants, protists that live in those freshwater systems that basically will pump that water out because remember, they want to live in that environment. And so therefore, again, just getting rid of that excess water itself. So again, we kind of look at the whole passive transport. I mean, you could basically provide so many different examples for this as far as oxygen, carbon dioxide. You've got water, you've got salinity, you've got sugars, you've got basically any of those type of molecules that we need. Now, kind of leading into the active transport, and here's where I kind of change the trend a little bit because we do have certain molecules that need to be forced into the cell, and that's where the active transport comes in. Energy is required, pumped in there. Okay, but I think where we also jump ship is we start looking at more along the terms of energy in the whole kind of concentration gradient. And that if you've got something that is in passive transport, it's working from high concentration or low concentration, right? That we start talking potential energy versus kinetic energy is that as those molecules are going through, right, and working to that lower concentration level, we start to see an increase in kinetic energy. As you are, in the form of active transport, it generally means that you're forcing something up the hill, up, to a higher concentration level. Well, the only reason we'd really probably want to be doing that in the future is obviously to be stockpiling it and hopefully harnessing energy that we can eventually use as it works back down that membrane system again. So, and it doesn't always work that way for everything. Last video, we talked about antibodies and how they can mark cells for death if they're bacteria and viruses. Well, we have cells called macrophages, macro meaning large, that they're going to come in and they'll eventually read that so-called signal, that mark, and they're going to basically take in those viruses, the bacteria, and eventually break them down. Well, how do they do that? Well, they engulf it. And so it's a form of active transport because energy is required. But what they do is they completely will surround that cell and eventually they will take in that virus or that bacteria and engulf it, break it down, and get it ready to be distributed throughout the body and however that happens. So we've got that going on. The ATP energy, obviously, it's required, to force something across. We didn't talk a lot about the sodium potassium pump and how our nervous system works. Kind of the same approach, that it's basically just stockpiling certain ions on the inside and outside and allowing that chemical reaction, allowing the electrical stimulus eventually to move down the line. But I think kind of where we also go with this is the whole process of cell energy and the process of cell respiration. And when you think about the active versus passive transport that's happening in that process, in that as the electron transport chain is occurring, remember what's happening is that energy is being released from the electrons, and that energy is being used to pump protons across the concentration gradient and actually work it on the other side of the cell membrane. Remember, there's a double membrane system in the mitochondria, in that you've got basically now we're stockpiling those protons up here. And as we're stockpiling those protons, we're actually increasing the amount of potential energy that eventually they work themselves down that little turbine that makes ATP, and eventually that potential energy is stored into or is harnessed into kinetic energy, transfers into kinetic energy, and we all of a sudden have energy that is used throughout the body. So I think, again, we could kind of go a couple different ways with that passive versus active transport. And that, you start looking at those materials that we need inside the cells and on the outside of the cells. But I think we also start taking in terms of energy as far as, where is energy is being released and where is energy being stored, and how are we utilizing that energy, and why would we ever want to use energy to create energy? Well, prime example again is in the cell respiration, in that process of the electron transport chain and eventually into chemiosmosis. So again, as we go through, we're going to eventually work into water potential, which again has a lot to do with the whole passive transport and basically the movement of water in and out of a cell, which is probably one of the molecules that moves easiest, just based on its size. Even though it is polar, it just simply can fit through the aquaporons that are produced there for it. So a lot of good things happen there for water, but just based on the environment. So we'll come back and learn again, just kind of revamp how to calculate water potential and what that means.